welcome to Market Open Direct Connect, a webinar series for companies to tell their story, why it's compelling and why it's an opportunity to invest. I'm your host, Stuart Walters, and we're joined here today by Dr. Liz Dallimore, the CEO and Managing Director of Argenica Therapeutics, ASX code AGN. Liz will provide a company update, run through the latest phase 2 data, and participate in a Q&A post-presentation. A recording of this webinar will be available on Market Open and Argenica's communication channels within the next 24 hours. So I'd like to introduce our guest. Liz, welcome to Direct Connect, and over to you.
Thanks, Stuart, and thanks to everyone online really for tuning in, and we're really sort of delighted to have the opportunity to talk through our phase 2 clinical trial data. We've seen a signal of efficacy in this data, which for us, in terms of the patient group where we seem to see a signal in efficacy and a 15% infarct volume reduction, is a really, really critical group for stroke care, and the most at high risk and the most at need patients in acute ischemic stroke, large vessel occlusion. So look, we are obviously the market reaction has sort of not been super favourable. We understand not hitting that very broad secondary endpoint has not met some people's expectations. We do reiterate that this is very much a safety study, phase 2 study. We're delighted that we have hit that primary endpoint, and we're really excited in terms of seeing that efficacy signal that we do see in this patient population. A phase 2 trial is very much about signal searching. So, really what we're trying to do here is work out, what we see in our preclinical animal studies, which are really comprehensive and have really shown that this asset is worth pursuing in clinical development. We're really trying to understand, does that translate to humans? And if it does translate to humans, what sort of patient group does it do best in? So we did have the foresight in this trial to make sure when we were analysing the data, we had a predefined subgroup around collateral. So there's probably a little bit to unpack there. So what I have done, because people are probably very sick of hearing from me, and you want to actually hear from the team that sits behind this amazing clinical trial and the amazing research that has happened. So I have brought onto this webinar Dr. Meghan Thomas, who is our VP of Clinical Development. Meg has been absolutely instrumental in running this phase 2 trial. It is not easy running clinical trials in acute stroke patients in emergency departments, and I cannot say just how amazingly proud and just so delighted with the work that Meg has done working with our CRO ProPharma and also project management team at Alethea Life Sciences. So Meg has so much to be proud of. She's pulled off an absolutely phenomenal feat in getting this phase 2 trial done, bringing in timelines, 12 months. We've had fantastic recruitment. We've had amazing take up from principal investigators at all the sites who have also done a fantastic job. So I cannot be more proud of the work that Meg and the team have done to actually execute on this trial and see something that is really, really positive in terms of this, not only the safety, but also the efficacy signal. And I'll also introduce Dr. David Blacker, who is the chairman of our clinical advisory committee and an extremely distinguished stroke neurologist who is much better positioned to talk to investors around the relevance of the slow collateral subgroup that we have seen this efficacy signal in. So I will hand over firstly to Meg, just to kind of give a bit of a summary on what we have seen in our phase 2 data results and just refresh everyone's memory in terms of what we actually did in the phase 2, and then we'll hand over to David, who will give more context around the collaterals that we see in these acute ischemic stroke patients with large vessel occlusion. So Meg, I'll hand over to you.
Thanks, Liz. Just wanted to say too, it's been a really amazing working with all the trial sites. They've been incredibly engaged, and they're all really excited to sort of be working with us on the next steps as well. But just to reiterate, when we first started this trial, we didn't know if our drug was safe in stroke patients. So, you know, in our phase 1 trial, we'd given it to healthy adults with an average age of, you know, sort of 36. And we know that in stroke patients, they do tend to be sicker, they are older, and there is more things that can go wrong in that scenario. So for us, it was really important to show that our drug was safe in a phase 2 trial. It's a little bit different to oncology trials, for example, where, you know, you're not trying necessarily to be safe. You are looking for that cell death. But in other indications, you know, safety is what usually causes a drug not to progress. So for us, looking at that was a really important scenario. So we're really pleased that when we look at those treatment emergent adverse events, and this covers everything that happens in a patient who has a stroke, we don't really see any difference between those that received the active drug versus those that received the placebo. So we feel that our primary endpoint of safety, the drug was safe and it was well tolerated. So that for us is a really important tick that we've achieved that. Just on this slide too, we're still digging into all of this data again, but we did want to look at whether we could see a difference between those people that received a thrombolytic drug. And so far we can't see any indication of that either. So again, that's another really major tick or pass for us. And then thirdly, would just our secondary endpoint, which was that overall infarct reduction, we didn't see a difference. But what we have done is we have tried to think of every single condition or factor that might impact that, and then we've gone off to have a look to see if we can see that sort of responder profile at all. And so, yeah, just on this one, we're really pleased that we've met our safety endpoint. As Liz has mentioned, we've found a signal that we think is worth exploring, and David will cover off on that a little bit more as well. But my final point to make is that I think we've done a lot of watching of others and how trials have progressed. And what we're really keen to do, we've been very clear about it, is you start broad. So, you know, we've included everyone who's had a stroke out to 24 hours after their stroke. So being able to show that our drug is safe in a really sort of every sort of condition that you come into with a large vessel occlusion, showing that our drug is safe in that broad population gives us a lot of confidence. And then the intention has always been to signal search within this trial and then narrow down that focus of patients going forward to really try and get that efficacy signal. So what we did, as most of you will be familiar with, is a patient has a stroke, they're frequently in an ambulance. They usually call a tertiary hospital as a code stroke. And so our clinicians at the tertiary hospitals were able to sort of start seeing or thinking about if that patient was going to be eligible. Once they arrive at hospital, they have their CT scan where it's confirmed that they have a large vessel occlusion stroke. And as soon as that happens, then they can be randomized and our drug can be administered. So anyone who is eligible for thrombectomy was likely to be eligible for our trial. And then they get the drug, they get the thrombectomy treatment, and then we follow them up out to 90 days and assess adverse events, infarct reduction, and a variety of quality of life measures as well. As Liz said, when we first started this trial, we anticipated recruitment to be between 18 to 24 months. So we were incredibly pleased that we were able to pull it into 12 months. And I think that is testament to the protocol design, where we worked really hard with David Blacker and the CAC team to sort of make sure that the protocol reflected standard of care as much as possible. We've, you know, engaged really well with all of the sites. We went to every single site initiation visit, and the feedback from the principal investigators was that the protocol was largely a standard of care, but they were also incredibly pleased to be supporting an Australian developed drug and to be part of an Australian clinical trial. So I think, you know, thanks to the PIs and the study coordinators at all of those sites, and to the patients and their family. So we managed to dose 92 patients at 8 of the 10 sites that we had open. As we've said, we're doing safety tolerability. Our pharmacokinetic work is still coming in, and we're looking for that preliminary efficacy signal.
Thanks, Meg. I think we sort of now move on to really in terms of looking at this slow collateral subgroup. So I'll just just explain before I introduce David that these were predefined subgroups. So we did have a hypothesis essentially that the collateral blood flow that we see in these patients could impact where we see an efficacy signal. And this is largely because other trials have seen a similar thing, especially with thrombectomy trials. Thrombectomy trials that add TPA, that patients that have good collaterals actually do well. So we figured if we're delivering a neuroprotective drug quite early on, the patient has good collaterals already, they're doing really well, the delta that we would see in the infarct would be lower. So that's why we had these really predefined subgroups. In terms of looking at the subgroup efficacy signals, we are comparing patients that have slow collaterals that have received our drug with patients that have slow collaterals that haven't received our drug. So we're really looking at the like for like, so that we're really comparing what is that sort of final infarct volume on slow collateral patients that haven't had our drug versus have had our drug, and what's the delta there. So that's really sort of what we're narrowing down into. David will talk through the relevance of the collaterals and how that it really is assessing these collaterals is actually standard of care. So something that is done anyway. So an easy thing to point to sort of in the next phase of the trial in terms of inclusion criteria. So David, I will hand over to you to sort of give a bit more colour from someone that lives and breathes this in their everyday life.
Thank you very much, Liz. I'd just like to reiterate some of the words that Meghan talked about. I can't think of another stroke study where we actually finished our recruitment more than 6 months ahead of the schedule. So it was actually quite incredible to have my colleagues from around the country all collaborating and cooperating. And there's a great sense of enthusiasm amongst them, and I'm sure they're all very eager to hear our results. The safety data is very reassuring, and particularly the fact that this is a quite representative standard sample of a stroke population with an average age of 72. Our phase 1 study of healthy volunteers was average age of 36. So you can see that we have a much more relevant now picture for safety. But just I think for the investors in the audience to back up a little bit to talk about collaterals, I might get you to advance the slide one further to show the CTs, because the CTs almost speak for themselves. The kind of stroke that we're treating here is a stroke that's obstructing the big blood vessels in the brain. Now, the way that the circulation of the brain is arranged is that there are 2 large arteries at the front, the circulation of the carotid, 2 large arteries at the back, and called the vertebral arteries, and they all join together at the base of the skull. And there is some anatomical degree of cross flow between those different channels that you're actually born with and designed with. But what that means is that sometimes you can block off one artery on one side of the circulation, and there's cross flow from another side that fills it in. As we age and as we develop atherosclerosis, there are small obstructions that appear in the blood flow. And it's a bit like putting a dam in a river, and little tributaries will start to open up. Now, if you acutely block off that river, the patients who've got good blood flow tributaries already established, if you can actually maintain blood flow through those tributaries, the brain tissue will survive. If you can't, the brain tissue dies. So we have a scenario where you can have a blockage in an artery, and there's still the maintenance of the blood flow, and the collaterals are very good, and those people are going to get better almost regardless of what you do. With the artery being opened, they've got a very good prognosis. So it's very hard to add to that. At the other end, you've got people who've got a blockage, and there's absolutely no tributaries opened up. That's what we call the poor or absence collaterals. So they've got a grim prognosis, whichever way we go. And then finally, there's this in-between group, which I was thinking is a bit like the Goldilocks zone. So it's a group of patients who've got a tissue that's underperfused, but if it just can survive a bit longer to open up the artery, the outcome will be good. Now, when we're actually seeing the patients first up, we don't necessarily know all of this at the time. So this is all derived from a post hoc analysis, and but it's very exciting to me because it actually makes a lot of logical sense. So I think this is a key finding, and I think the more and more I think about it, this remains a very large group of patients. Up to 30% of people fit into this zone of intermediate collaterals, and that adds up to tremendous potential benefit. So in a way, this trial has achieved everything we'd hoped for, pretty much. It's demonstrated safety, and it's given us an efficacy signal. And that's what phase 2 studies are all about.
Thanks, David. I think that's a really good overview, and that kind of that Goldilocks zone that we see where we're seeing this efficacy signal. So we know that patients with those good collaterals that we don't have as much penumbra there because they're getting sufficient blood flow to keep that part of the brain alive. And one of the things, looking at understanding the mechanism of action of ARG007, working on exocytotoxicity, working on oxidative stress, working on mitochondrial dysfunction. These are the things that are heightened in these slow collateral patients because they're not getting that blood flow that they need to that part of the brain. So it would, definitely be an adjunct. I know there's a lot of questions coming in. So I think what I might do is sort of just go through the last couple of slides, and then we can actually spend quite a bit of time on the questions because I think they're really pertinent. It's really important for people to understand what this means in terms of designing future trials and our sort of thinking on how we can get the delta even bigger in this patient subgroup, but also reiterating that there's a lot more data to come in. So we haven't done any post hoc analysis yet. This is all on the predefined groups that we had already built into the trial, which I think is really important. Post hoc will give a lot more colour. But we are unblinded, right, when we're doing post hoc. So really important that, seeing this efficacy signal was not from unblinded post hoc data analysis. So, just sort of reiterating the point around, so what is clinically meaningful? A 15% infarct volume reduction, it is a really, I think, important clinically meaningful signal. So really what we want to do is we want to bed that down in the next phase of the trial. So pulling in that target patient population. As I said before, measuring collaterals is standard of care. It was done in every single patient that we had in this trial. As David said, the 30% of slow collaterals, it's really reflective of what we see in the large vessel occlusion stroke patients. So a really good, a really sort of good understanding of that broader patient population and where we think the drug is going to work. So just want to reiterate a couple of the, key points that came out from the announcement this morning that, really what this phase 2 has done. And as David has said, it's done exactly what we needed it to do. Primary endpoint is safety. One of the other things that's really important about safety is that, showing the drug is safe in that broader stroke population is really important if we want to deliver this drug earlier. So we did try to build in this phase 2 the ability to actually dose patients out in, say, a regional hospital that they were then transferred into a thrombectomy centre, because we knew that would give the drug more time to actually protect against that growing infarct. From an ethics point of view, we weren't able to do that because we had no safety data on this drug. So we couldn't reasonably dose a patient in a rural or regional setting and then have them transferred in if we didn't know whether the drug was safe or not. So really important point to make. This showing this safety actually now gives us further options around that clinical trial design going forward. And seeing a signal of efficacy in this subgroup where we had already hypothesized we thought that collaterals may impact is an absolute massive tick in terms of what we wanted out of this phase 2. So reiterating both Meg and David's point, we're so delighted. The PIs have been absolutely exceptional. Meg and David themselves have put so much work and thought into not only designing this trial, but the execution of the trial. And I, again, can't thank them enough for all the work that they've done. And I think, just again, reiterating the point, our preclinical data that we have on this drug asset is so compelling. What you do in animal studies is you have the ability to manage variables. So, you've got animals that are all of the same age, all having the same occlusion, all having the same follow-up. So because you're really trying to distill down what is that treatment effect. In a phase 2 trial, we do have a lot of variability in stroke patients. So we've tried to minimise that as much as we can by making sure we're only having large vessel occlusion patients, looking at sort of aspect score, and again, then looking at the collaterals as well as a key part that we're looking at that may impact treatment. So it really sort of goes to show, that we are proving the concept of the ability for this drug to work in stroke patients in humans. And lastly, we are really well funded. So 7 mil cash in the bank as of today, plus an extra 3.5 to 4 mil of R&D tax coming back as well. So we'll have 11 mil coming in. The costs for the trial are pretty much paid for. So we've got good cash balance to give us the opportunity to start reviewing all the data that's still to come in. One of the really key parts of data that we're looking forward to seeing is the pharmacokinetics. So where blood sampling has been done in stroke patients to understand, have we got the dose right? We're seeing an efficacy signal, but with potentially a different dose. If we map that all onto our preclinical studies, could we actually get an even bigger signal by looking at another dose? So we're really excited as a company. We're absolutely delighted how the trial was executed. We've got a safe drug and now a drug that shows an efficacy signal. So it's really exciting things to come for Argenica, and it's a good day. So we're really pleased. So Stuart, I'm happy to hand over to you and see. There's a number of questions, so we'll go through it.
Thanks, Liz, David, and Megan. Fantastic presentation, and congratulations on everything you've achieved so far. There are a lot of questions that have come in, so we'll get straight on to those ones now. Liz, I'll ask, how does the company view this result in terms of the market opportunity?
Yeah, so the market opportunity is, now really 30% of, that broader large vessel occlusion patient group. So that's sort of sitting probably at around 10 to 11% of that large vessel occlusion group. So if I look at a comparator, the sort of nearest comparator that we have is alteplase. So that also targets about 9% of the stroke patient population, ischemic stroke patient population. So it's probably on par with a thrombolytic drug market. So that drug market is projected to be $3.8 billion by 2030. So this is still a massive market, but probably one of the things that we haven't unpacked there is once we actually drill down to show that this drug is working within this subgroup. So these are the highest risk, most at need patients that have the worst outcomes. There is no reason, given that we know that the drug is safe, an ambulance arrives to a stroke patient. You won't know if they have good collaterals, poor collaterals, intermediate collaterals. But if the drug is safe, and if you get that drug in earlier, the patients that are highest risk of the worst outcomes will have a better outcome if you get their drug. So you will be delivering that drug to every stroke patient still. So from a clinical development point of view, we do have to, now really drill down into the question, is is the drug really working in these stroke patients? We'll power the study better and look at ways that we can actually increase that delta as well beyond 15%. So still a massive market, even if we were just to go for the slow collateral group for sure.
If you thought ARG-007 would work better in slow collateral patients, why didn't you only recruit these patients?
That is a really good question. So again, it comes back to, clinical trials are experiments, right? You're asking questions. So the first question we wanted to ask in this trial was, is this drug safe in every large vessel occlusion stroke patient? So to answer that question, we have to go broad and we have to get all large vessel occlusion stroke patients, regardless of collaterals. And then the second question we want to ask is, does it work the same across all large vessel occlusion patients? So does it work as well as it does in rapid collateral patients that we know have good outcomes as opposed to slow collateral patients? So to actually design the clinical trial properly with good scientific experimental rigour, we had to include a broader patient group.
And just to add to
Sorry?
Yeah.
when
Yes.
you don't know all this information, and it's coming on stream very quickly, and you don't want to preclude a patient from the standard therapies at that point. So in the future, we'll be able to become more selective. But when you're in the acute setting, provided the indications and contraindications for standard use and for the trial have been met, you just go ahead, and then in the washout, and then analyze those particular subgroups.
And just leading on from that, I think some of the other questions around, looking at thrombolytic drugs or not, we've all got a lot of questions about how this drug works in this population. Does it work in patients who receive a thrombolytic or not? So if you ask just one question, you're only going to get one answer. And so the idea of this study was to, again, show that it's safe, like David said, in that broad population, and then hone in on which group is responding or not. So I think, we've been able to we will be able to look at all of those things as more of that data comes in.
It's almost like a concertina. We start broad and then we bring it down. And then once we've proven it in that smaller targeted population, we can then spring it out back again into the bigger group.
How does the efficacy signal the slow collateral subgroup influence the design of any future clinical trials for ARG-007?
Yeah, so this is now really the subgroup that we sort of want to narrow in on. So the design of the trial, we're collecting collaterals anyway as standard of care. So, and knowing it's safe now, we can bring on some of those regional hospitals as well. So where they're undertaking an assessment of the collaterals as well through perfusion imaging. So that allows also then to get that bigger delta. So the patient will have the drug earlier. They get transferred into a thrombectomy centre. But if we can sort of focus on this trial. And Stuart, I did just want to, go to a question where, an investor has sort of said, if the intent was to only focus on this subgroup, if you thought it was only going to work in this subgroup, you should have sort of notified that of the market. So that was a hypothesis that we had, but everything in our preclinical studies shows that the drug works more broadly. So we are still learning quite a lot about thrombectomy patients and collaterals. So it was a hypothesis. So we were obviously hoping that, we'd see some sort of effect broadly, absolutely. So, which is it's essentially what we see in our preclinical studies. But, that we did build in this subgroup in case we sort of had this, where we did start seeing these good collaterals coming out. And again, like I said, this is all sort of evolving based on the learnings that we're learning about thrombectomy and the types of patients and better imaging modalities. So unfortunately, it's just the nature of science, things sort of churn. But absolutely, of course, we wanted to see that bigger effect on the secondary efficacy endpoint for sure. So I don't think it was misleading to tell investors that, really that's what we're hoping for. I think we've always been very clear that this was a safety study and a signal searching study first and foremost. Absolutely.
Liz, can you explain how even modest reductions of infarct volume translate to meaningful patient outcomes in terms of independence and recovery?
Yeah, so I've had a slide on that. So a lot of work has kind of gone on around these thrombectomy trials to see, what a good infarct volume reduction is. So we know the minimal clinically meaningful reduction is around 1.6%. That comes from a meta-analysis of these thrombectomy trials, but we're probably really looking around that 5 to 10%. So yeah, we're sort of sitting above that, but again, obviously we'll confirm this efficacy signal in a sort of 2B type of study.
Given the notional effect size you've seen here in the slow collateral subgroup, any indication on the sample size you'd anticipate in the next potentially pivotal trial?
We have no idea. No, it's way too early to tell. And that takes some very, very clever statisticians to really to take all our data, look at, essentially how it's working, where it's working, and then build that in also through looking at sort of preclinical models and other data sets to get to that sample size.
As a technical follow-up, perhaps could you describe how the patient's collateral assessment is performed at the point of enrolment? Perfusion scan that's already standard of care?
There are a few programs that are being implemented at hospitals now that sort of overlay AI on those scans. So as well as clinicians being able to make that call, there are also programs, Branomics and Rapid AI are two examples that provide that sort of response. David?
Yeah, so just backing up from that, the way it works is that as soon as you're in this the CT scan suite, the dye and contrast is ready to go, and there's set protocols. It all happens quite automatically. If you step back, when this started 15 years ago or thereabouts, it was very crude. You just look at the delay in dye appearing on one side of the brain versus the other. And now with the automated computer programs and the advent of AI, it's a very exciting time where these technologies are going to be able to quantify things much more accurately, and we'll have a much better way of looking at it, even at several different time points.
Were patients categorised into subgroups prior to receiving the treatment with ARG-007? And is the 15% infarct volume reduction compared to placebo of the same subgroup?
Yeah, so I mean, the way that it worked is we weren't selecting patients based on their collaterals, but we were analysing the data based on collaterals. So it was a predetermined group of analysis based on those collateral groups. So when the patients come in, we're just making sure they've got large vessel occlusions, they're eligible for thrombectomy, and we had a few other sort of inclusion criteria. So because we didn't want to, necessarily skew it to a particular collateral status, we wanted to understand, is the drug safe across all patients as a representative group of large vessel occlusion patients, essentially. So definitely not selected, but it was predefined. So not post hoc analysis. It's done. The analysis is done based on that collateral status, which is data that's collected as part of our electronic data capture process.
With this new signal in the highest risk patient groups, does it streamline the design for your next clinical trial?
Yes, absolutely. So yeah, having a subgroup patient population almost gives us a much better thing to grab onto to design, a trial that, hopefully is going to show, an efficacy signal based on what we already know. Having that proof of concept already in humans allows us to really target it down into that patient group. So predetermined subgroups is a really attractive way to do clinical trials to give it the best chance of success. Also something that the FDA likes, the more you can kind of predefine your patient population, and narrow it down, the more they like those sort of trials.
Isn't it the case that by the time that a subgroup patient is identified, it's too late to give the treatment?
No, because we did that in this trial. So we had all that information about as you know, David was saying, in current standard of care, the collaterals are being looked at anyway. So it's an important piece of information to understand, you know, what's happening with that patient. So, you know, we would give it at the time of collateral identification. So as I said before, what we can do now with a safer drug is we actually can bring in, we don't have to do the trial necessarily in a thrombectomy centre now, so we can do it closer to where the patient is at the time that they're having stroke, so that we're protecting the brain for longer as that patient comes into thrombectomy. So there's a lot of benefits to actually now narrowing down this group and having that safety information as well.
What would the next trial look like? Is it going to be challenging to diagnose a subset of stroke patients to then dose them, thus delaying the speed that they can be dosed? Yep.
No. So standard of care, you diagnose collaterals as standard of care anyway. They're doing it out at hospitals. Collaterals are a really key part of understanding what's happening with these stroke patients. So understanding what sort of stroke they have on imaging is really important. So do they have an ischemic stroke? Is it caused by a clot, or is it a hemorrhagic stroke? Because that really changes the treatment that those patients get. And then obviously looking at how well is that brain perfused? What is that blood flow look like? Where is the location of the clot? How big is the clot? All those sorts of things. That is information that is being gathered on every stroke patient that's coming in anyway. It will not delay it. It didn't delay it in the current trial. So yeah, it's a non-issue.
Were there signals of clinical improvements beyond radiological changes?
So not that we have seen. So the subgroup analysis was done on the secondary endpoint only in terms of looking at the data that we've got now, but we will dig into that post hoc analysis and see within this particular group if there's anything else interesting that we're seeing.
Can you comment on the possible importance of administering ARG-007 well before reperfusion? So if it was administered, say, in an ambulance well before the patient gets reperfusion, is it likely the drug will also provide benefit to patients other than those that had a positive effect in the trial?
Yeah, absolutely, possibly. Yeah, I know. So we know that the earlier the better generally, but going to David's point around, you know, the collateral status, we do know that good collateral patients, you know, they do better because their brain is perfused. The higher delta that we're going to see is on these slow collateral patients. So if you imagine, you know, that infarct growing over the time, the penumbra turning to infarct over time, especially in these slow collateral patients, the earlier you give it to protect the brain, the bigger delta you'll see with a placebo group. So yeah, we believe that if we can do it earlier, we'll see a greater effect.
The trial looked at efficacy signals at circa 3 days. Preclinical work looked at 28 days. What is the significance of time or more time in the efficacy signals? Yeah,
so the preclinical work, I think that question is referring to as our non-clinical primate data. So in that study, we actually looked at 24 hours and 28 days, and we don't see that much of a difference. So we typically in stroke trials look at final infarct volume. So that's pretty well defined. So anywhere between that day 3 plus or minus a day, we know that that's a pretty good indicator of that final infarct volume.
Assuming success in future trials for the poor collaterals patient population, would the expectation be that frontline medical staff use ARG-007 on all ischemic stroke patients, as you know the state of collaterals, until you do a scan in hospital?
Yeah, absolutely. So if you have a drug that is safe and you know that it works in the highest risk patients that have the worst outcomes, you are going to give that drug to everyone, right? So if you're picking up a stroke patient in the ambulance and you don't know the collateral status, but you have a drug that can potentially, you know, reduce the growth of that infarct by 15% and hopefully more that we can establish in future trials, you're going to give it to all patients because it's safe. So it's similar, I guess, to what they do with heart attack patients, giving patients aspirin. We know it doesn't actually, do much in patients that have good collaterals following heart attack, but it works really well in patients that have poor collaterals. So, a similar kind of analogy that someone gave me, a clinician gave me this morning, that I thought was quite a good analogy.
Does only working in a subgroup of patients change the total addressable market for the company? Would this affect who you have the drug to in practice?
Yeah, and I think I sort of spoke about that a bit in the presentation. So, what this probably more akin to now, if we just deliver it to slow collateral patients, is very similar to the thrombolytic drug market, the TPA market, which is projected to reach $3.8 billion US dollars per year in 2030. So it's still a big market. If it is given to every stroke patient in an ambulance still, obviously that changes the market size again.
Is this a commercially attractive outcome and subgroup for big pharma?
Yeah, so we believe so. So I'll be picking up discussions with pharma and get them across the results. So the pharma companies that I've already had quite a lot of discussions with, in terms of, we know that a 15% infarct volume reduction is clinically meaningful. So, yeah, absolutely. And we'll continue to have those discussions.
And does the outcome of the safety and efficacy assist with other indications such as HIE and TBI?
Yes, very good question. So safety, absolutely. So, it's a big tick. We've got safety in a really compromised group of patients, in a neurology setting. So for TBI in particular, the other thing that, we can see that it's sort of working really in that acute phase. So with moderate to severe traumatic brain injury patients, the majority of them actually come into hospital within a 2-hour window because there's some sort of trauma that has obviously happened that's caused the traumatic brain injury, like a motor vehicle accident, a fall, for example. So these patients actually do come into hospital a lot quicker than stroke patients. So if our drug is really working in that acute phase and getting it in sooner is better, then this does lend itself to looking at that opportunity in TBI for sure.
And did you see any other subgroup differences between TPA and non-TPA group within 3 hours, within 6 hours versus between 6 to 24 hours, etc.? Also, given the results, do you think you would explore ARG-007 in non-thrombectomy LVO AIS patients as well?
All good questions. So, Meg, I don't think we've done that post-hoc analysis. So, we got this data sort of early Saturday morning. There was a coding issue in it. We had to send it back to the US. We had to jump on a call, our midnight Saturday night to go through it again. So it's been a whirlwind, probably 48 hours of going through the data. So we don't have definitive, we didn't sort of pre-specify, hours from TPA and all those different sort of nuances of different groups and pulling it together. So we will look at all of that in post-hoc, but this collateral, it was the really clear signal that we saw out of the trial that was, really that sort of standout. But as I said before, Meg and the team will be doing a huge amount of post-hoc analysis to give more colour to the data.
We did look at timing from onset to when people received the drug, but we haven't combined that yet with collaterals. So we've done individual sub-analysis around time, we've done an individual sub-analysis around collaterals, but we haven't put those two things together. It's a pretty small, relatively small phase 2 trial. So the more permutations you do, the less numbers you end up in each group and the less informative it becomes. But that is definitely some of the work that we're looking to do, or just to sort of really dig through all of those questions to see what impact it might have. But like Liz says, we've not had the data for that long yet, but that is definitely on the board of things to dig into and see if we can see any signal in that.
Is it possible to assess a patient's collateral flow at the time of the stroke, or do you just administer the drug regardless?
Can I just maybe change the conversation a little bit on this one? I think there's two questions, right? So what do we need to do to get the drug approved? So we need to show efficacy in a group, whatever that group is. But and so designing a trial to get that efficacy signal is really important. But the second part to that is what would happen clinically. So if we can show that the drug is safe and we can give it to anyone with a large vessel occlusion, then clinicians will do that. And then off the back of the label, you know that it will be approved for a particular subgroup. So I think we're trying to, you know, there's the two questions that we really want to address. So showing it's safe is actually quite exciting. I know no one gets excited about safety, but it actually gives you the ability to do a lot and clinicians comfort to deliver your drug to a wide variety of patients. And then what your drug's actually approved to do on the label is very much about trying to show the level of efficacy you can achieve in a particular group. We might see lower levels of efficacy in other groups, but because we're really trying to get that approval, we're really trying to weight it towards a group that we see the most amount of efficacy. Does that, Liz, does that kind of make sense?
I think that's a good point. I mean, drugs are often used what we call off-label, right? So what you're trying to do in the clinical development path is work out where does your drug work best to give you the best chance of seeing efficacy in a particular patient group. So, and you don't want the noise. If we in the next stage bring in good collateral patients that we know don't have much of a delta on infarct, don't have much for our drug to work on, why would we bring those in when it's going to dilute the effect of the slow collateral patients? So we keep it at that. Once a drug is approved, you have clinicians that will use drugs essentially off-label. So this has been happening in stroke for quite some time with a licensed drug called tenecteplase. It was safe and it has been used in heart patients, so patients with a particular cardiac issue. Had not been approved for stroke, but there was an idea that it would work well, similar to TPA, and there were some additional benefits. So clinicians started using it off-label. So the label was not approved to use it in stroke patients, but the drug was deemed safe, and so clinicians started using it. So we see a similar sort of thing would be happening. If you know that your drug is safe, you know that it's going to have a benefit to the highest risk patients, you deliver it in an ambulance before diagnosis of collaterals.
And then just to add to it, as time goes on, that delta in the good collateral group that you can't prove to be significant in a small sample size, as you then expand it to more and more people, you then can demonstrate significance. So, like I said,
exactly.
It's a Jenner effect. We need to narrow it down so we can go forward.
Yeah.
Once we've proven that, we can then look more broadly.
Another one here is a 15% reduction really compelling, say compared to Aptol or NA1?
Yeah. So look, so NA1 didn't see an infarct volume reduction on their second phase 3 trial, nor did they see it in their Frontier ambulance trial. So I'll sort of just make that point. Obviously, they've got an issue with the interactions with TPA for their drug. So Aptol did see a 40% infarct volume reduction, but we don't really understand. So there was no statistical significant difference within that either. So they present on confidence intervals similar to what we do. So we don't really understand exactly if there's a particular sort of patient population that they've looked at in that. We you know, I guess don't have as much data, but 15% clinically is really significant. So if we can hone in on that and get more data and more confidence that, you know, that effect is real, give the drug earlier to increase that delta, then that's what we'll be looking to do in the phase 2 clinical trial for sure. But yeah, absolutely, really clinically meaningful, that 15%.
I got a question here for David. Could you talk about the importance of dwell time for a neuroprotective agent like ARG-007 before thrombectomy, and how would you really reduce the variability there, especially if it's not a regional transfer?
The whole concept of dwell time is really related to pre-hospital processes, ambulance transportation, and efficiency of your service. And for a long time, those factors have been a disadvantage in places like Western Australia, where you've only got thrombectomy capability in Perth, and you've got patients coming from a long way from distant country areas. But almost paradoxically, that gives us a chance to study that angle a little bit more, because if you're in, say, a big US centre or European centre, the neuroprotective drug might only have 10, 15 minutes to get in before the thrombectomy is achieved. Whereas if we can get this rural sites to administer it, we might have several hours of impact before the thrombectomy is done. So when we look back at our original goals, that whole concept of expanding that time window of reducing brain vulnerability and slowing down the cascade of ischemic related neuronal death, we're looking good for that.
That's an important point around sort of other jurisdictions. You know, typically stroke care works on this sort of hub and spoke type of model, where you have a hub, which is a thrombectomy centre, and then you have all the spokes that come off it, which are sort of stroke care units, and that's sort of quite prevalent in the US as well. So, you know, I think broader sort of opportunities, you know, beyond sort of how our healthcare system works in Australia, but looking, say, to the US in how it will work in delivery around sort of stroke care units, and still having transfer patients coming into that hub thrombectomy centres in those sort of large metro areas as well.
Could the drug be effective in strokes of lesser severity, that is non-LVO strokes?
Yeah, good question. So, I mean, we've got preclinical data in permanent stroke models, which essentially means that the occlusion is not removed. We went for large vessel occlusion strokes for a number of reasons, because they have the biggest infarct, so we, you know, expected to see the biggest delta on these patients. But yeah, you know, potentially we probably need to dig into that to look back at our preclinical data and see if there's something there. We do know now that thrombectomy doesn't work in the, you know, like an M2 vessel occlusion, you know, so probably need to look at thrombectomy trials as well, what they're sort of seeing and where we think the drug will work.
Do these insights help inform the studies for other indications, TBI, HIE, etc.?
Yeah, so I kind of ran through that before. Absolutely, from a safety point of view, that is a massive tick. And also, you know, the fact that we kind of, through this, we've sort of really distilled down that mechanism of action in terms of that acute ischemic cascade. So what we see in traumatic brain injury is also this exocytotoxicity, this mitochondrial dysfunction, and so those patients are coming into ED a lot quicker than stroke patients. So typically within 2 hours, as I sort of said before, that they, you know, there's some sort of trauma, there's something that's happened that people witness, and so the ambulance does tend to arrive quickly.
Now that the data has been released, can you follow up with the patients that responded positively to the drug to see its longer-term benefits?
Typically not, for data privacy reasons. You don't tend to sort of do that with stroke trials. So we do have 90-day follow-up data from those patients. So that was part of the exploratory endpoint. So we can definitely look into that data of the group that seems to be responding.
What do these results mean for the previous discussions re potential partnering?
Yeah, so look, I will go back to the pharma partners and walk them through, obviously, the data, the results, and see, you know, is there an appetite to potentially fund a phase 2B, knowing that we can really hone in and do a good trial design, honing in this particular patient population, and see if there's appetite for partnering there. So those discussions will definitely continue and be ongoing.
Given the potential importance of early administration of the drug in future trials, you need to know if the stroke is ischemic or hemorrhagic before administration of the drug?
Yeah, so, well, again, that comes down to safety, right? So that was the whole point of sort of proving safety in ischemic stroke patient population, is to say, well, you know, we don't sort of exacerbate bleeding, etc. So showing that it's got a good safety profile in terms of those treatment emergent adverse events is really important to then allow us to actually go into ambulance. So Yeah, look, we're coming up to the hour, Stu. I'm sure there's a truckload more questions. People are absolutely, we've got our investor hub. I answer all those questions on the investor hub. So we probably, unless there's anything completely burning there. But, you know, I just sort of want to reiterate that, you know, yes, we didn't sort of hit the secondary efficacy endpoint. We'd really hope, based on our preclinical studies, you know, that we would get there, but we are really, really excited about seeing this efficacy signal in this subgroup, who are the patients that actually need this neuroprotective drug the most. So, you know, the good collateral patients, they have good outcomes. These low collateral patients, they have really poor outcomes post-stroke. And if there's anything that Argenica can do to help these patients and, you know, really focus in on efficacy within these patients, you know, we're really keen to do this. And we are just so delighted with how this trial has been run, the people that have been involved, how quickly the recruitment's happened, showing that the drug is safe and showing this efficacy signal. There's huge amounts of positives in this, and look, we're really excited about next steps for the company.
Liz, that was echoed in the Q&A tab. There's a lot of great comments that weren't read out there. So congratulations to you all on everything you have done so far. That is all for today. There's some great questions, and we really appreciate you guys taking part in the Q&A and also for the presentation earlier. For more information about Argenica Therapeutics, head to the company website, argenica.com.au, or you can follow the company's social media channels, or like Liz said earlier, you can also join up to their investor hub. Once again, thank you to the three of you for taking part today. To our attendees, thank you for logging on. Some great questions, and we look forward to chatting again soon.